Okay, so hello. We'll start this first parallel session, session one. And this parallel session will have guest presentations. Today is from, I hope I pronounce it right, Shiladitya,
Yep.
Banerjee, and his student Logan Carpenter. They'll be talking about the modeling in CompuCell that they did for the eLife paper in 2001. I just mentioned brief mention that Shiladitya has a he works at the cellular level and the subcellular level, look at cytoskeleton, single cell, and collective behavior of tissues, of cell behaviors in tissues. And remember that this meeting is being recorded and will be uploaded to YouTube. And with that, I will just hand it over to Shiladitya and to present his nice work. Thank you. Thank you for joining us.
Yeah. Thank you very much. Thank you, Julio, and thank you, the organizers, for inviting us to share our work on modeling cell computation using CompuCell3D. So at first, I want to say that we are not experts in CompuCell3D, but we used it for some of our recent work, and we found it really easy, user-friendly, and adaptable. So I think that this talk is an example of how you can start using it without any prior knowledge. So this talk will be co-presented by myself and Logan Carpenter, who is a PhD student working in my group at Carnegie Mellon. And this work was done in collaboration with the labs of Guillaume Charraz at UCL, who led the experimental side of the work, and Alan Lowe, who developed deep learning methods to segment, track, and determine cell-fed dynamics, which we used in our model. And our former PhD student at UCL, where I was 2 years ago, and Daniel Gradici, who led the computational part of the work. So most of what I'm going to talk about has been published in this eLife paper in 2021. So this is the outline of this module. And first, I'm going to discuss what is cell computation, give you a brief, sort of very basic background from biological perspective, what is cell computation and what we know about it, and how we can experimentally characterize cell computation dynamics, right? Then I'm going to go through the method for developing the computational model, like what are the underlying physical components of the model, and how we use experimental data or information to build a model, right? And then I'm going to show you a working example of the model in CompuCell3D and how the model compares with data. And then there will be a few simple exercises with the model that Logan will lead. And we have designed 3 to 4 exercises that you can easily do to get a feel of the model, okay? So at any point in time, if you have questions, please feel free to stop me and ask questions for clarification, okay? So yeah, what is cell computation? So cell computation is a quality control process that aims to remove less fit cells from a tissue. So one of the main hallmarks of cell computation is that it's context dependent. So let's say we have a clone of cells with genotype A, right? Which is viable when situated among cells from the same genotype, right? However, when you mix this mutant genotype A, let's say with wild type cells, and if the wild type cells are fitter than, let's say, this genotype A, then the wild type cells can remove the cell type A by competition, okay? However, mutation can also sometimes give rise to more aggressive phenotypes. For example, we can get super competitive cells, right? So like cancer cells, which are fitter than wild type cells. In that case, what happens is that the super competitors can kill off wild type cells by competition. So it's highly context dependent. So it depends on, what the relative fitness of the cells are. Okay, so cell competition was first identified in developmental biology studies back in the 70s. So it was initially identified in minute heterozygous flies, the flies that where ribosome synthesis was affected. And if these minute clones, which are shown in blue here, when they're in contact with wild-type cells, they get killed, okay? They get eliminated, and in red, you see cells that are dying, which we call apoptosis, and apoptosis occurs at the interface between the normal cells and these minute clones, okay? We can also imagine other types of genetic modification. For example, here you see at the bottom row cells that are more aggressive than wild type. For example, this MYC mutants, which are cells with a higher level of oncogene, right? And these cells can grow and actually can replace, eliminate fully wild-type cells. Okay, so you can see that at the end of the experiment, there are no wild-type cells left. Wild-type cells here are shown in pink, right? And because this aggressive mutants can actually kill off and eliminate these wild-type cells, right? So these are examples of what we call a fitness fingerprint or biochemical computation. So here computation is occurring at the interface between two cells, and the extent of contact between cells determine the probability that a cell will die, okay? So the neighborhood of a cell is very important here. So what you can see here is overall the tissue shape and topology doesn't change much, but what happens is that over time, the more aggressive cell, which I call winner here, takes over the entire tissue by competition, okay? So this is called biochemical competition, which is short range and contact based. There is another type of competition which was discovered in actually 5-6 years ago. It's called mechanical competition, okay? And mechanical competition relies on the concept of homeostatic density. So here the concept is that each cell line that you see has a preferred density that it prefers to reach, okay? So if the density, for example, is higher than that preferred density, then those cells will get eliminated, so you can reduce the density to reach your homeostatic density. So what you see here in the top row are two cell types, a fast-growing population of cells in pink, and a slow-growing population of cells, which is surrounding the fast-growing phenotype in green, right? So as the fast-growing cells expand, like they grow and divide rapidly, more rapidly than the green cells, they actually exert mechanical pressure on the green cells. So the green cells get compressed, right? So this compression creates a high density, okay? And because of that, the cells get extruded, okay? And so this is called mechanical competition. So, so competition can result from two types of mechanism. One is mechanical competition, which can arise from differential growth. Like you mix a fast-growing population of cells with a slow-growing population of cells, there could be differential growth. And this differential growth results in pressure buildup. And the pressure leads to increase in density. And the rate of cell elimination increases with the density, right? So this is shown here in cartoon because there was no quantification available before we came into the field and studied it. So I'm going to go through that in a bit. But so this is mechanical competition. The other competition that I talked about before was the biochemical competition, where the rate of elimination of cells depend on how much perimeter is in contact, okay? So cell competition actually then can be formulated as a balance problem, right? So the net growth of a cell, right? So the survival of a cell depends on its growth rate, and the growth rate depends on the rate of proliferation of the cells, like how fast the cells are dividing, minus the rate of elimination, like how fast the cells are dying, right? And both these factors are not constant parameters. They depend on the local neighborhood of the cells, mechanical forces, etc. Right? So the problem is very complex at the single cell level, right? So I hope I've been able to convince you of that. So what we want to understand is how do cell level dynamics control tissue scale population makeup, right? So we need a sort of a modeling strategy that can handle this multi-scale problem, okay? So because events are occurring at the single cell level, and they are leading to shifts in population makeup, right? So before I talk about modeling, I want to talk about the experimental side of the thing. So how can we say something more quantitative about this phenomenon, right? And so depending on various factors, such as the arrangement of the cells, the initial cell count, etc., you can expect different types of outcomes. Like you can expect that the two cells that we make can lead to coexistence, sorting, or elimination, right? So what is the outcome that we see? So, this project, when it started, we started by doing experiments. So these are experiments done in Guillaume Shiraz's lab. So this is an example of competition between MDCK epithelial cells and MDCK cells with Scribble gene knockdown. So what Scribble gene does, so knockdown of Scribble gene leads to two main effects. The Scribble mutants grow more slowly, and they have reduced adhesion, so they don't adhere to other cells that well. Okay, so let me start the movie again. So we start with a 90-10 ratio, okay, of the wild type cells to Scribble, right? And what you can see here is the number of cells with green nuclei, right? These are the cells that over time they get eliminated. Okay, here you see that the green cells are the Scribble mutants, right? Because the nuclei is level, and when they come in contact or they're under pressure, they get eliminated and killed off by these wild type cells, right? So you can actually image this population and actually can calculate things, okay? So that's what Alan Lowe's, our collaborator, we segmented the cells, looked at, so these are actually competition assays where we labeled the nuclei of the two cell types, right? And then we track the cells. By tracking the cells, we can compute various things. Its position, its the number of cells, local densities, etc. All sort of dynamic properties. And also we can compute things like probability transitions from one state to the other, right? So what is the rate at which cells are dying? What is the rate at which cells are growing, etc. Okay. So using such techniques, automated techniques, which is long-term single cell imaging and tracking, we were able to get data such as this, which is an example showing cell count versus time for a competition assay based on 90% wild type cells and 10% mutant cells. And what you see here is that in green, you see the cell count for the wild type cells. Cells actually grow, they divide and reach a homeostasis over time, which means that they reach sort of a constant cell count. Now, homeostasis means that the system is not static, but there is a dynamic balance between rate of growth and death, okay? And by contrast, the mutant cells quickly die off, okay, because they're eliminated by computation. And what you see here is, aside from getting data on counting, we can also get data on what is the cumulative count for cell death, right? So you can see that, for example, the mutant cells are dying at a much faster rate, okay? Two important data that we need to know, because we will include that in our model. So as I said, that the probability that the cells are dividing is not a constant, and it depends on the local density of the neighborhood. So for example, the probability of death for the MDCK wild type cells, we see that it decreases with density. This is not very surprising. This is a phenomenon known as contact inhibition of proliferation. So if cells are in a crowded environment, they grow more slowly, okay? The opposite is seen for Scribble cells. We don't really understand this origin. The mutant cell, for example, has more like a non-monotonic trend. It sort of increases its division when it's in high density neighborhood, okay? So this is a surprising result that we see from the data, right? And when we look at probability of apoptosis, we see very different behavior for the mutant versus wild type. The mutant cells are more sensitive to crowding, so they die. So the probability of apoptosis or death for the mutant cells are much higher than the wild type cells at a similar comparable density, right? So you can see that there will be competition between these two cell types because they have different rate of growth and different rate of death. So we can first approach this problem by simply writing down rate equations, okay? And you can ask, why do we need to use CompuCell3D or use POTS model to model this? We can first actually approach this using simple rate equations. We know the rates, we know the density, we can predict what happens. I'm not going to go through the details, but I just want to show you that we did that, and we are not able to capture, for example, behavior of cell count or density, etc. Okay, I don't want to go through the details of the equations, but I just wanted to show. I just wanted to inform that the simple rate equations models don't capture what we see at the population scale. We are not able to capture the dynamics of cell count, etc., with knowing the rates. And the reason is these continuous models or continuum models at sort of population scale miss one important thing that we need, okay? So we need a model that can model cellular level phenomena, right? What we see in the experiments in the local neighborhood of a cell is very important, okay? And this cannot be captured in such continuous model where we write down rate equations, right? So we looked at, for example, maps of division. So this plot is a bit complicated, but it shows the probability of division of wild type cells when it is contact with n number of mutant cells and n number of wild type cells. What we see here is that the probability of division depends on the neighborhood, right? So when wild type cells are surrounded by more mutants, they divide faster, okay? And on the other hand, the probability of apoptosis is also elevated in loser cells, okay, when they are surrounded by other cell types, okay? So, so these are the type of results that cannot be easily incorporated in continuum models. So we decided to use cellular parts model, okay? And we built on and as I will discuss in the next few slides, so we construct a physical model of competition that accounts for not only this long-range type of density-based density-based competition, but also short-range signaling where cells can sense each other's neighborhood and can induce death or induce faster growth, okay? So, so here's the framework of the model that we use. So as I said before, we are not experts in CompuCell3D, but we wanted the model to look something like this. And we saw that CompuCell3D gives us a very flexible platform to adapt the model to what we need, okay? So we wanted to create a multi-layered model for computation, which has two layers, a physical layer, where we compute deformation of the cells, cell size, cell shape, right? Its mechanics, its movement based on physical forces, right? And physical forces will be depending on sort of elastic forces, adhesion forces, etc. And that physical layer will be coupled to a decision-making layer, right? And in the decision-making layer, cellular decision-making is implemented at each time point as a set of probabilistic rules that we determine from experimental data, right? So that this mechanical model is coupled to a model where we have probabilistic rules, and those rules are inferred from data. So what I showed you so far with experimental data are very important because we want to directly use this information in the model, okay? So I'm going to show you how we do that. Before that, let me briefly describe the physical layer of the model, okay? I think there are some questions in the chat. Let me quickly address the question. So is this compartmental model like in COVID? I'm not familiar with the COVID model that you're talking about.
Well, the set of equations seem to be, what is it called? Nested. And if there is no immediate answer to it, you can skip my question. I may be wrong, I may be ignorant too.
I'm actually ignorant about the COVID model you're talking about. So maybe the
compartmental model is very popular, and you also were talking population levels. So
yes, right. So in that sense,
but you left the differential equation system in favor of Potts model.
Yeah, because that
left it, so it doesn't matter now. Those equations don't matter.
Yeah. I just wanted to show you that those equations can describe sort of the global macroscopic properties, but it misses a lot of important details that are happening at the single cell level, right?
Exactly. So Potts is much better.
Yes. And I'm going to discuss more about it just now.
Thanks for the clarification.
Yeah, no problem. Next. So if you have a question, please shout out because I'm not looking. This is to everyone. So next question is from Bharat Vivan. So I was wondering, how do you know they are undergoing apoptosis? Very good question. So you can look at the nuclear morphology of a cell and the nuclear morphology of a cell. So for a dying cell, the nucleus actually disintegrates, okay? And And then we
Yeah, right. But is that the only thing you've checked for? Did you do some sort of tunnel assay or something to specifically tell that it's apoptosis, or it's just based on your nucleus getting disintegrated?
nuclear getting disintegrated and the cell shrinks, and it just becomes it just dies.
Sure.
Okay, and then
but I'm saying it might not be signaling involved. It could be that the other cells which are proliferating faster are just mechanically pushing it, and it just gets slightly lifted off from the substratum. And because it doesn't find anything,
of course,
open, right?
Of
So
course.
I'm not sure if it's like signal mediated apoptosis or it's happening due to mechanics.
Right. You're not letting me finish. So, okay. So what I'm trying to say, yes, it can happen. Cells can get extruded. And you can also get signal-mediated apoptosis. We literally see signals of apoptosis, like when the nucleus disintegrates and breaks up, right? That you don't see in extruding cells. Extruding cells just disappear, right?
That's right. That's right.
Those things happen. But we don't know what you're right. We don't know whether it is mechanics based or whether it's contact signaling based, right? But from the sort of the neighborhood maps that I just constructed from experimental data, I showed you that the rate of apoptosis depends on the neighborhood, right? It also depends on density and mechanical.
That's right. I just had this additional question if you checked for it or not. Okay.
Yeah. So we checked for it, and then. And then there was a whole process of training a machine learning algorithm to identify these things. Okay, so
thank you. Yeah, that explains it. Thank you.
Yeah, great. Okay, so first of all, the physical layer of the model. So let me try to describe it. So the physical layer computes cell deformation based on mechanical properties. So here I used cellular parts model. So I know that since this is the sort of last day, fourth day of the workshop, you are all familiar with cellular parts models. So I'm not going to go through the details. I just want to say that what we essentially do here, we determine cell shape and size by minimizing a Hamiltonian for the parts model, right? And Hamiltonian has two parts. The first part is adhesion energy with between cells, right? And the second part describes penalty for cell deformation if the cells deviate from the target area too much, right? So here are some key parameters of the model, right? So each cell has a deformable, sort of an elastic modulus, lambda, which characterizes how easy it is to deform the cells, right? So each cell has the elastic constants lambda and lambda 2. They will be different for the two cell types, okay? And I'm going to come back to it later on. This is very important. And secondly, there are intercellular adhesion energies defined by these parameters J, right? And there will be homotypic adhesion, cells of the same type adhering to one another, and also heterotypic adhesion J12, okay? In addition to the two cell types, we have a third cell type, which is the free space, okay? So the free space or the substrate is treated as a third cell type in the model. And we have to account for adhesion energy between the cell and the substrate, right? So cells that have a higher spread area, right, they will have a higher adhesion with the substrate, okay? So then we use this parts model. And then how do we couple it to the decision-making layer? So here's how it works. So first we calculate cell shapes based on mechanical and adhesive property, and also motion vectors. So we also have, each cell has a motion vector. I'm not going to talk about it because it's not very important for this presentation. But yeah, so basically you calculate cell shape and motion based on energy, minimizing energy. And then there is an automaton layer where you make fit decision for each cell based on its area, density, or other properties, okay? So so once you calculate the cell shape, you then assign probabilities for whether the cell will grow. Growth means you change the target area of the cell. So this is something important. So each cell has a target area. If you deviate from the target area, it will cost you elastic energy, right? So you basically grow by increasing your target area. So there's a probability for that. There's a probability for dying, and there's a probability for dividing. These are not constant rates. These are rates that I showed you. They depend on the neighborhood of the cell density, etc. And I'm going to discuss them right now. Any questions so far? Okay. Okay. So First of all, let me describe simply the laws, the rules for cell growth and mitosis, division. Mitosis means cell division. So each cell has a cell cycle. And it is known that MDCK epithelial cells, they grow at a linear rate. They grow linearly, not exponentially. Other cell types may grow exponentially. Okay, it's not very important here. But what is known here is that the MDCK cells grow by adding a fixed volume per division cycle. So in each division cycle, they grow by adding a fixed cell size. Okay, so this is called the adder model of cell size control. So each cell has a birth size, it adds a fixed volume, grows at a rate, and then divides. Okay, so this is the cell cycle model. Okay, and more importantly, this is the growth law. So each cell grows at a rate G, right? This is the target area of the cell. Okay, we also have to worry about contact inhibition. Okay, what that means is cells can grow freely at a fixed rate when it is isolated. But as cells divide and gets crowded, it becomes more difficult for cells to grow due to lack of space and due to this effect of crowding, right? And this is modeled by this equation. So there is a parameter K, which determines how sensitive you are to contact inhibition, right? So as K is high, then the rate of change of target area decreases with density, because if you are in a very dense neighborhood, your target area is much different from your actual area is much different from your target area, right? So that means the rate of growth decreases, okay? So then we model this something called contact inhibition, okay? So these are the rules for growth and division. And then we couple it with the rule for apoptosis. So as I said, that there are two different types of apoptosis that can happen in this computation. So one is mechanical, where the probability of apoptosis depends on the local density of each cell. So each cell has a local density. And for example, in the two mutants that we are working, so in the computation assays we are working with, the mutant in red, which we call the loser cells because they grow more slowly. So this is just a terminology, winner, loser, right? So loser is something we call they grow more slowly, less fit, okay? The red cells have they are more sensitive to crowding, so more sensitive to increase in density. So they have a probability of apoptosis that is different from the winner cells, which have a much higher threshold for crowding. Similarly, there is a biochemical computation, which is signal-based apoptosis, as one of you asked. And then, so the probability of apoptosis in that case depends on how much you are in contact with your neighboring cells, okay? So if the percentage of heterotypic contact increases, probability of death increases, right? So these are the two mechanisms for dying. So how do actually cells die? The cells die, so once they meet this rule, meet this criterion, then they're eliminated by setting their target area to 0. So we set their target area to 0, right? At that instant, then they shrink, and then they get extruded out, okay? And then it is seen in experiments that the cells can also get extruded, okay? So it's called extrusion, live cell extrusion, as one of you were just saying. So in live cell extrusion, what happens if the cell area decreases beyond a certain threshold, right? They don't have enough adhesion with the substrate. So they don't like to be in the monolayer anymore and they get extruded, okay? So we sort of assumed, this is an assumption, we don't really know what is the threshold, but we assume that cells get extruded when the area is less than half of the average area, okay? But this is something we can adjust in the model pretty easily, okay? So once a cell is committed—
I have a doubt. So the model which you built is 3D, right? You're placing it on ECM.
No, this is a 2D model.
Okay. So there is no ECM on it.
No, so ECM is the free. No, there is no ECM layer, but the free space is the ECM, we can say.
Okay, wonderful. Thank you.
Thank you. So once a cell is committed to extruding, it is eliminated immediately from the tissue. Okay. So in summary, what we have is a sort of a multi-scale, multi-layered model, physical layer where we compute cell deformation, coupled to a decision-making layer where you have these probabilistic rules, okay? So let me show you now an implementation of the model in CompuCell3D. Okay. So first I'm going to show you just an output, and then we're going to compare it to experimental data. And more importantly, I want to talk a bit about parameterization of the model, which is very important because this model comes with several parameters. Okay.
So these rules, these
Yeah.
mechanical and biochemical rules,
Yes.
are part of the program of apoptosis?
Yes.
Because when I learned about cancer and cells, they were talking that cells are programmed to commit suicide under certain conditions. Are these the conditions?
This is what we found, yes, through experimental data and analysis. So we
Okay,
looked at
perfect.
dying cells, and we looked at what are the characteristics of those dying cells, like what predicts death, right? So this is a very difficult biology question, very fundamental. What predicts death? So we saw that there are two properties. One is the local density, how compacted they are, right? And also the other is the contact with the other mutant. Okay, so these two things. So here I'm going to show you an example of mechanical competition. So there is no biochemical competition implemented in this simulation. Logan will talk about biochemical competition later on. So this is just a simple simulation that I'm going to show. Here, the initial seeding density is 1:1. So we will have 1:1 ratio of, so this is the initial configuration, 1:1 ratio of the red cells and the green cells. The red cells are loser cells. They have by loser, I mean they have less fitness, they grow more slowly, right? These are the mutants, and the greens are the wild type cell, okay? And now, as I play the movie, first, just let's look at the movie. I'm gonna go to the data in a bit. So as you look at the movie, the cells are growing, expanding, they reach confluence. You see these blue and yellow flickers. Those are apoptosis events. Yellow is the apoptosis of mutants, and blue is the apoptosis of the wild type cells. So let me play it again. Okay. So what you see here, what is clear is that the mutants get eliminated pretty rapidly, right? Although they start at a similar ratio, the mutants get eliminated, right? So you can see the population count, which is the top left curve. The red cells decrease very fast. And you also have the green cells, which are reaching a homeostasis by eliminating the red cells. The green cells reach a homeostatic density and an average cell volume, right? Constant. More strikingly, the apoptosis count in the red cells is way higher because they are more sensitive to crowding, okay? So that's the key difference here. So what is setting in competition here is that the red cells are much softer, they can get compressed, right? And as they get compressed, they are in a more crowded environment, and they're also more sensitive to crowding. So this mechanical difference sets the competition here, okay? So this is just an example, and the model is very general. You can change the rules, and you can study different types of scenarios. So it's a very simple one. Let me discuss more about the model now. Okay, so this is very important. I think for anybody trying to build a model, one has to think about where the parameters come from, right? So there are some physical parameters of the model, like, for example, what is the target area? And the target area is calibrated by looking at spread area of single cells in isolation from experimental data, right? So these are experimental data from our collaborators who actually looked at the cells, and we got this area, okay? Great. The adhesion energies are calibrated such that we get the correct cell shape, okay? And how do we define cell shape? Cell shape can be defined as the ratio of perimeter to square root of area, so that's one thing. You also look at what is the coordination number of each vertex, etc. So by looking at topology and shape, you can actually calibrate the adhesion constants. And by looking at the ratio of cell size, like relative area of each cell type, right? We can actually calibrate their compressional modulus, okay? So these are physical parameters. Then you have to specify the initial conditions, which you know, right, from the experimental data. We actually ran similar experiments. So we know cell count, cell position, cell size distribution, and cell type, okay? So then you have to fit it to the model. Importantly, the model has different rules, as I was talking about. The growth rule, right, how the cell area evolves in time. We took that from data. Division rule is something we measured, right, by looking at probability of division versus probability of division versus local neighbor density, etc. We also looked at apoptosis rules. Extrusion rule is taken from experimental data. Contact inhibition is something it's not possible to measure really, okay, in experiments. So that is sort of an adjustable parameter in the model, okay? And then we fit the model, and then we sort of ran the model, and we get certain outputs, right? And so I think one of the parameters we fixed is contact inhibition. And I just want to show you, so this is a success of the model, and also like CompuCell3D, that we can actually capture very accurately evolution of cell counts. So what you see here, the top row here is the model. The bottom row here is the experiments. We can accurately capture the cell count of the two cell types. The green one is the winner cells, and the pink one are the loser cells. These two cells are competing. And I said what regulates competition here is that the mechanical competition between these two cell types results from the combination of two factors. The pink cells have a lower stiffness, which leads to more compression and compaction, and because they have higher sensitivity to crowding, they get eliminated fast. Okay, so the model can capture very well features such as density, cell count, etc. And we did several things with the model, studied different contexts. Okay, so what Logan will now go through are some simple exercises that we implemented with the model, where we want to show you how it can capture different scenarios which are biologically relevant. Okay, I can take questions if you have, or
yeah, I missed the beginning, the introduction, the few minutes of introduction because of the technicalities. So I want to decipher exactly the word competition. Who is competing with whom? And also over what? Space, nutrients, fighting with the mutants, or they're all losing to the mutants automatically?
Yeah, so here what I'm showing you, Logan will just share his screen. So here what I'm showing you is competition for space is one thing. And the competitions also happen between mutants, right? So what you want to do is you want to maintain homeostasis, right, in a tissue, and that happens through, so basically we're looking at competition between cells of different growth rates, different fitness, and who can win, okay? Yes, Pratik.
Yeah, hi. Thanks for the next talk. So you mentioned that say the loser cell has lower stiffness compared to the winner cells. So how do you bring in the aspect of stiffness in CompuCell3D? Is it through energies or?
Yeah, so in cellular parts model, there is an energy Hamiltonian that determines cell shape. And one component of that Hamiltonian is area compressibility. Right, so in this 2D model, you penalize changes in cell area from the target area, right? And the parameter that describes the cost of that energy that deformation is what we call the stiffness. Okay.
Okay.
So the cells are very soft; they can deform more and deviate more from the target area.
Okay, understood. So one more point, like maybe I didn't understand properly. So you mentioned that apoptosis can be the mechanical based or the bios, the signaling based.
Yeah.
So, but in reality, are they independent or it's
Yeah.
or what I mean is like they cannot be totally independent phenomenon, right? So
Excellent question. So I think one will influence the other, right? But I don't think, I mean, they're independent in the sense that the origin is different, right? So the mechanism is different. So signaling happens to ligands when cells are in contact. Mechanics doesn't care so much about contacts like close contacts or actual contacts. They can happen over longer range. So these are different mechanisms. But one can influence the other. Like they can act in synergy or they can be antagonistic to one another, you see? That's a very important question, but yeah.
okay, thank you.
Thank you. Logan, yes, go ahead.
All right. Julio, quick question. We're still finishing at 4:15 Eastern time. I'll take silence as a
Yes,
yes.
I think so.
Okay, just checking.
Because it was some kind of delay, I think all the parallel sessions will delay a little bit, so it's probably okay if you go over 5 or 10 minutes, no problem. Okay.
All right. Well, so we're going to be going over 3 exercises. Exercise 1 is cell competition and tissue homeostasis. Exercise 2 is mechanical stress-driven competition, and exercise 3 is the interplay of mechanical and biochemical competition and the role of tissue organization in a cell. Let's see. Okay. So just a warning at the top, and that's that I program like a physicist. So you may wonder, what does this mean? And it means I get the job done, but it isn't necessarily very pretty. But by all means, if you see any non-optimized code or bad practices, feel free to send me any complaints, suggestions, or compliments. And you can send that to log@cmu.edu. So as Sheila was talking or saying, this exercise will be addressing tissue homeostasis for a single cell type. And this will help us get a better understanding of the parameters that we're including and considering. Like this tissue just did, we'll be able to, we define homeostasis by that dynamic state when the rate of division and death is balanced. First, we'll run a simulation that starts with a single cell and grows to reach homeostasis. For this run, density death rate will be set to true. This means that the function over here on the right is used to calculate the probability of death. If you're feeling adventurous, you can change these values here, which are the parameters for that function, and can change the shape of the curve. But keep track of any changes that you make because you'll want to carry them over to exercise 2. In the second part of exercise 1, the density death rate will be set to false. And this will change the probability of death rate to just be a constant probability. And we'll set this value here, P apo const, to be a value that you think is maybe too high. What is too high? If it kills all the cells, you've chosen too high of a value, so just a high value. You can feel that out through experimentation, and then we'll do a too low value, and there's not really too low of a value, so go nuts. And after that, you'll compare the homeostatic densities for these simulations, and we'll be doing that just through visual inspection of plots that are generated during the simulation. And down here in this snippet, you can see that there are several booleans that you can set to true and false, which determine which plots you'll see as the simulation runs. And yeah. So we were just talking about some of these global parameters that are up top, and there are more that are below this, like stiffness and contact inhibition, that you can, depending on how comfortable you feel, you can edit those as well. Just keep track of which ones you edit. And below all of those globals, there are the class definitions, which we'll go over briefly just to understand how they work. So first, we have the adder model initializer, and this is called once at the very beginning of the simulation. And here in the start function, we're calling set initial cell adder parameters, which is this function here. And it loops over each cell. And we'll see this kind of distinguishing cell type. If it's cell type 1, which is a wild type cell as we define it, then it gets assigned wild type values. Globals. And then if it's cell type 2, which is a mutant as we define it, it gets the mutant values. And then its adder mitosis condition is saved in a global list called adder condition list that we'll see again in just a second. In crowded growth steppable, the purpose of this class is to implement the increment in the target volume. And in the start is just plotting parameters. And so I collapsed that because it doesn't matter for this. And the orange dots indicate where the target volume is actually being calculated. And so we have a growth variable G that's determined by the cell type and the contact inhibition, and then the actual calculation and incrementation of the target volume. And now we have an adder mitosis steppable, and similarly in this one we loop over all of the cells in the cell list, and then for each cell we grab their entry in the adder condition list, and we check in to see if its current volume minus its birth target volume is greater than the added volume for mitosis. And we just make sure that it's not been programmed to death, which is cell type 3 and 4. And if this is the case, it's added to the cells to divide list, which then afterwards is looped over, and we have a typical cell division process for CompuCell3D. Here we have the density death steppable. And the purpose of this is to calculate the probability of death based on density for all the cells. In this particular exercise 1, this version of this class, there's just a little bit extra right here to be able to switch back and forth between the constant rate of death and the density death dependence. But other than that, it's the same. So starting back up here up top, we loop over all the cells, and we check to make sure in these two lines that the cell isn't too close to the edge, because then we can get artifact effects of the boundary. And then we make sure that the cell's not dying. And if these are the case, then what we calculate, it's a local density based on the neighbors around it. And then based on the cell type, there's the random probability of whether it will die. And then if that's true, then its cell type is set to 3 or 4. Its target volume is set to 0, and it's given a relatively high stiffness so that it collapses and dies. Last, we have extrusion. In we have the cell, we calculate the average volume for mutant and wild type cells, and then we loop over all the cells and check if their volume is less than some threshold ratio or fraction of that average. And if so, they're immediately deleted and removed from the simulation. Okay, so now everyone can, I'll be, well, let's see. So right as this talk was starting, I went to run the program, and I realized that I left an error in. I fixed it, but I guess I didn't save it before closing the program or the editor. And so if everyone pulls up their exercise 1, so the name of the folder is CC3D Workshop Exercises Summer 2022. I probably should have thrown in cell competition into that title. Everyone can see my screen, correct? With the folders here.
Yeah, we can.
Okay, just checking. I wasn't, okay. So in exercise 1, single cell homeostasis, open that up and go to homeostasis steppable.py. And scroll down to line 62. And the error is right here. Wild type total div count editable. Delete the second editable.
I'm sorry to interrupt. Was this shared? I don't know where to find this. Can you just guide me through fast?
Yeah, I can put a link
Sure,
in the chat.
that'll be great. Thank you.
No problem. So there's the link. So yeah, once you delete that second editable typo, you're good to run. And to show, so I guess I'll do that a little bit slower. So under file, click open simulation file.cc3d. Navigate to wherever you've stored the folder, the exercises, and click on the homeostasis.cc3d within exercise 1. Open, and then run. If you want to change any of those parameters, they're here within the homeostasis steppables file. I presume the silence means it's going well.
Can you repeat the thing to change? You said there was some mistake. We're supposed to change that.
Yes. So it's down in line 62.
Uh-huh.
It should be, as you see right here, wild type total div count editable, but I accidentally
sorry.
had, yeah, the second
Yeah.
underscore.
Yeah.
Yeah.
Got it. Thank you.
Okay, so mine just finished running. It took 3 minutes and 20 seconds. Now, I'll try setting it to something too high. I can't remember what I did before, so I'll try 5%. It seems too high. Maybe 2.5%. Oh, that was done. I need to set this to false. Now it'll be using this constant probability of death of 2.5%. Okay, maybe that's too high. One percent.
So, how are people doing with the exercises? Do you guys need more time? Is there anything that Logan can help you? If you need more
Yeah,
time, you can send an anonymous message in the chat. No problem.
I was just gonna say we should probably move on to, instead of running the last one with too low of a probability, should probably move on to the next exercise. And yeah, because we're kind of approaching, well, we have 15 minutes left for the official end time. All right. So
Okay, at least one student got it to work, and she's looking forward to the next. Right, so
that's code for the first one was boring. I get it. The next one's better, don't worry. Okay, so this is an example output for the default parameters, and the final simulation looked similar to this. And so the correct way to do this, of course, is to record the data, and then you average over several simulations to create an average local density, an average of the average local densities. But here we're just going to eyeball it, and this kind of seems to hover around 0.034. And we notice that with the too high probability and the too low probability, the constant probabilities, they seem to be approaching the same homeostatic density, which tells you that so long as a less competitive mutant type is viable, meaning it doesn't die immediately, the homeostatic density is approximately the same, at least just for this very simple toy model case. So now we're going to do a exercise based on the mechanical stress-driven competition. And in this exercise, we simulate experiments performed in Eisenhofer et al. From 2012, where cells were grown to confluence on a stretchable substrate and then subjected to a step deformation in one axis. As the deformation decreased cellular apical area, there was a sudden increase in cell density above what its homeostatic density is. And this resulted in a combination of apoptosis and live cell extrusion. And it's supposed to be on this one. And this led to a gradual decrease in density towards the initial homeostatic density after about 6 hours. So now we'll recreate this experiment in CompuCell. And so first we'll run the default parameters and allow the tissue to reach homeostasis, and we'll take note of that homeostatic density that the tissue approaches. Then we'll compress the tissue by mechanical force and resize the lattice. And then it will allow the compressed tissue to reach homeostasis again. So the parameters, and the global parameters up top will be largely the same, except now there's going to be 3 more. We have resize time, compression duration, and X force. So below in this little graphic, first in phase 2.1, we allow the tissue to reach homeostasis. And then in phase 2.2, compression begins, and there's a external potential that's turned on, and the strength of that is set by the X force parameter, and it lasts for as long as compression duration number of Monte Carlo steps. So in this case right here, 200. And then at Monte Carlo step of resize time, here 650, the lattice is resized, and in this new size, or in this new lattice size, the tissue is allowed to then reach homeostasis again. So we define a new class, compress and resize simulation for this. And here's a quick snapshot of what it'll look like at once the cell colony initially reaches homeostasis. Okay, so in this class, at the very start of the simulation, it builds a wall around the perimeter, and then compressed duration, number of Monte Carlo steps before the resize time. This wall that it made is destroyed, and a potential is turned on in the x direction with this cell.lambda vec x. Then at the resize time, that potential is set to 0, and the lattice is resized to be just bigger than the space that the cells are taking up in the x direction. And then the wall is rebuilt after all of that. So in order to use some of these functions, we need a few changes needed to be made in the XML file. A new cell type had to be defined for the wall, and then the adhesion energy between the wall and the other cell types had to be defined. And then to turn on an external potential, we had to import that plugin. And then a side note, if you go digging down below into the probability of death function, it's so for version 4.3 of 0.1 of CompuCell, the neighbor tracker app that is used for the density probability of death with a dependent on density function, and so it has some bug with resizing the lattice. So I had to put together a really hacky version to get this particular exercise to work. But there's a bug report submitted, and so that'll be fixed hopefully relatively soon. So this is what you'll see after the compression starts, or sorry, when the compression is finishing, and then the window will resize and we'll let it approach a homeostatic state. All right, so let's run this one. We'll go to File, Open, go to where you're storing these exercises. Exercise 2, compression homeostasis, compression.cc3d, and run it.
Hmm. Hmm.
That's a good question. So the question is, why do the cells get these shapes during compressing? Are these holes artifacts of the simulation? I'm not sure. As far as the exact shapes of the cells go, because I'm not familiar. I mean, I haven't looked at the pictures for the experiment. The holes that you see are extrusions. And so I mean, extrusions, live cell extrusions do happen. So I don't think that those are unphysical. But yeah, so I guess that's the answer. If that is that satisfying of an answer? Okay, now we're coming up on the end of this simulation. So have to cut the simulation short at around a 1000 Monte Carlo steps. But were we to continue to let it relax into a homeostatic state, we would see that it goes down and reaches the same homeostatic density as before with those default parameters from exercise 1.1. Oh, nice. Okay. The other sessions are also behind, so feel free to go over 10 or 15 minutes. Now, that'll be good. So we'll be able to at least do the first part of exercise 3. Otherwise, it was just going to be left as an exercise for the watchers, readers. Okay. So in this exercise, we look at mechanical and biochemical competition separately. So we'll model a mutation, I mean, that makes the cell less competitive. And you can see that through these choices in parameters. For example, here the growth rate is a little bit less. And the stiffness of the mutant cell is less, meaning it's more easily deformed. It takes up more space. It needs more volume in order to divide. Its outer volume is larger, and it's more likely to die from increases in density. So first, we'll create a fully mixed cell population, which will be initial case set equal to 1. And we'll turn off biochemical competition by commenting out these lines in competition.py. Then we'll run the simulation and take note of the final cell population counts. In part 2, we'll turn on biochemical competition and turn off mechanical competition, just the reverse of above, and then run the simulation again. And for part 3, which there most certainly won't be time for, we'll repeat 3.1 and 2, but for the other initial cases, which are 2 and 3, which are partially sorted and fully sorted.
Just a quick clarification. What is extrusion threshold? What does it imply?
So do you recall before
The area?
Yeah,
The fraction of area which is attached?
Exactly. Yeah, so it's
Okay,
this
okay.
fraction.
Cool.
Like Sheila said, we can arbitrarily choose it. And we go between a factor of 1/2 or 1/4 typically.
Cool, sure. So if basically it goes below that, your target volume becomes 0.
If it goes below that, the cell will be immediately deleted.
Right. Cool. Thank you.
Mhm. So these global parameters here define what the initial setup of the simulation is. For case 1, you can say what ratio of mutant to wild type cells you want, and the total initial cell count. For case 2, the mutants are in the little colonies, and you can define how many colonies you want, up to 9. And then for the case 3, they're fully mixed and they start on either side of the window. And this fraction here determines how much of their half of the window they take up. If it's 1, they're in the middle touching. If it's 0.5, as it is right there, then they each take up half of their half, so a fourth each. And then you have these parameters that we just talked about. So the differences for the class definitions are, well, so there's a different adder model initializer and a contact depth steppable, which is the biochemical competition. This is there's too many changes in here to really go over, so you can look at it on your own time, but this just determines which initial case is set up. In contact depth steppable, the purpose is to implement this probability of death function, where P is the percentage of the perimeter of the cell that's in contact with a cell of a different type. So that's accomplished in this loop. You go over each cell, and then in this collapsed for loop, it takes up a lot of space, so I had to collapse it for this code snippet. You calculate the neighbor, the cell type of it for each neighboring pixel, and then you calculate this function and probabilistically decide whether the cell will die or not, which is the same steps as before. Okay, so this is the exercise statement again. So we'll go over to CompuCell, open exercise 3. Let me just make sure. That's set to the, yeah, that looks good. Okay. And I should mention, as Sheila said, the correct way to do this, but the correct way to set these parameters is to look at experimental data and fit them as well as is possible. But these aren't those parameters fit to data. I chose these based on what would be what would run in a timely manner for these exercises. So yeah, this doesn't represent a real cell. It's a toy model. Oh. Okay. The other thing I should have highlighted, in order to switch between which kind of competition you're doing, you come into competition.py within the simulation folder of exercise 3. And in this case, I commented out, I have the density death steppable commented out and contact death steppable being called. And on my computer, I know that the contact death steppable runs in about 10 minutes and the density steppable runs in about 2 to 3 minutes. So letting these go will, for 3 point, like I'm doing 3.2 at this moment because it's the biochemical competition. Will take up the rest of the time. And while we're sitting here, if anyone has any questions, feel free to let
Yeah,
me
I was
provide.
gonna ask. Yeah, I was gonna ask in the contact apoptosis, is it line plot or a dots plot? Oh,
You mean here?
because it's moving in steps. I'm not sure why that is happening.
So in this particular step, there were several cells that died. So it
So
just,
if I say that,
incremented.
right, if I say at MCS 200, I have 30 cells dying, and at MCS 201, it stays 30. Does that mean that 30 new cells died, or is it the same number?
Same number, yeah, just a
Oh, I
cumulative
see.
run,
Okay.
total count, yeah.
Cool, got it. So it will not decrease basically.
Yeah, I mean, yeah, you could plot that, the number of cells that died rather than the cumulative.
Sure.
But yeah, in this case, it's just a cumulative, yeah.
Cool. I would have checked it, but it's just too long to check which plot it was. Thank you.
Yeah, no problem. And in my simulation here, the wild type one, pretty fast actually. I don't think I've seen him win this fast before. So no need for me to continue that one. Into switch. I go to competition.py and switch which one is commented out. And then run that. So yeah, I guess the moral of the story is CompuCell is pretty neat, and a really powerful tool. I believe the the paper that Sheila and his collaborators put out, or that was published last year, was the first time that there was a computational cell scale modeling of a competition system like this. So we look forward to the future or the coming years, as more groups will be adopting this method, and we'll be able to learn more about competition systems. And yeah, I'll run through what
I have a question in the chat. Maybe I can elaborate. I don't know if it needs to elaborate. Whether the processes are also competing in a way about who will kill first or who will make homeostasis still existing.
right. So
Sounds
you imagining
a
a
complicated
system?
question, no?
Yeah. If you could imagine a system where mechanical competition for the wild type cell is better at killing the mutant cell, but the biochemical competition of the mutant cell is better at killing the wild type cell, there could be some equilibrium and coexistence between the two where they're able to reach homeostasis. And this is a question that we're interested in trying to find an example and answer to.
I think people in ecology playing this kind of games a lot.
Mm-hmm. Yeah, certainly. Yeah. All of the systems of the cellular systems that I'm aware of, they both go together. If you're better at mechanical competition, you're also more fit in biochemical competition. But it's a very interesting question. I agree. Oh, that's fantastic. Someone's planning to implement this for a hackathon. I love to hear that. So here's two example outputs of the simulations I just ran, and some for the partially sorted, which initially start with little colonies, and fully sorted. And the fully sorted have the most difficult time resolving. They take a longer simulation to have elimination or not. And also fully sorted most favor biochemical competition due to the reduced surface area. And yeah, so have fun. And oh, someone just asked, what is the Mathematica file that I shared? That's just to help you see or visualize what the shapes of the probability curves look like for the biochemical and mechanical competition for the mutant and wild type cells relative to each other. So like the mechanical competition probability of death looks some, two logistic curves, where the mutant in this case, as I defined it, is more probabilistic than the wild type. But I just find the visualization helpful. So I just inserted it. But yeah, thanks for coming. And I hope that all of you can use this in a hackathon in the future, or research, either way. And yeah.
Thank you very much.
Thanks very much, Logan, for your exercises, and some people already be using the hackathon, so I'm very happy that we have this outcome. This is a signature of successful parallel session. So I guess the other parallel sessions are finished as well. I guess you will cover back in the news, the final module of the day at, I guess, 16:45. I'll check with the other organizers. And I guess if you still have questions for Logan, I don't know if you can stay here for the next more 5 minutes. Okay, but thank you very much. And I will stop recording right now. Okay, thank you, Logan.
Yeah, my pleasure.